Thank you, Jim. Thank you for the opportunity to ONT for presenting. I had a chance to meet Jim in our lab a few months ago to share what we're doing, and I'm excited to share this in this venue and with the audience online. No disclosure. So I'm going to talk about a very thin layer of tissue that is probably the most important thing in our entire body, and I'm not biased because I'm a retina specialist, but it is the retina, because it allows us to appreciate the world as we do. And as you can see, it's made up of a bunch of cells that are in a very standard format. And you can imagine any genetic disruption can lead to disease. And this disease can be devastating because it robs someone of their sight. It could be someone as young as someone who is just born, or as we get older due to different genetic elements. What we want to understand, though, is how can we better understand these retinal diseases, and ultimately, what can we do about them? The field of retinal disease is actually pretty young in the sense that for the longest time, we used to think these were autoimmune diseases. It wasn't until 1990 when there was the first understanding that there was a genetic component when rhodopsin, or the photopigment in the rod photoreceptor, was actually identified as causing retinal disease. Then, within 10 years, we've developed the first gene therapy for a different gene, RP65, in a dog model. The next breakthrough in this field wasn't unique to ophthalmology, but it really opened up the doors, and that's next-generation sequencing. And on the plot on the bottom, what you can appreciate is that we went from have a few genes that might be related to inherited retinal disease to now about 300. That number's plateaued off, and what we now understand is there's not likely more genes out there that are causing retinal disease, but they're gene variants in those diseases that are driving different phenotypic presentations. What's unique about ophthalmology is that we've been able to really move this area forward with genetics, and in 2017, we received the first FDA approval for gene therapy injectable into the eye. It was the first gene therapy for any organ system. And a few years later, we've had our first clinical trials of CRISPR-Cas9 correction of a different gene, CEP290, that was not amenable to gene augmentation therapy. So as you can see, the field has moved pretty fast. The downside is the genetics of IRDs are very heterogeneous in that we cannot look inside of a patient's eye and tell you what gene or even variant might be at play, and this really kind of limits our ability to drive a genotypic to phenotypic understanding. What really allows us to do that is genetics and understanding the variants that are driving disease. Now, in an ideal system, what we envision is that we're able to identify a mutation, and then we can drive the next step of therapy, which is design of elements to actually augment that gene product, whether it be silencing, augmentation, replacement, and ultimately, as we've shown, deliver this into the eye. But all of this depends on step one. Without gene mutation identification, we cannot proceed to the next few steps, and we're at a bottleneck. Now, disease variant identification has kind of evolved over time from single-Sanger sequencing to now gene panels that are the most widely used technique. In our clinic and many clinics around the world, after good clinical diagnosis, the next step is to send off genetic testing. And there are a few flavors that you could do. You could send off a targeted panel, which relies on short-read exome backbone, whole exome sequencing, or whole genome sequencing. The most common method is going to be targeted panel sequencing. And this uses an exome backbone using short-read sequencing. And as you can imagine, if you have a variant in gene 1 in an exon, you're going to find that variant, especially if it's a small nucleotide variant. But intronic variants on different genes are going to be missed, unfortunately. And actually, large-scale studies from our universities in the US and UK have showed that 30 to 40% of patients with inherited retinal disease do not get a clinical diagnosis or genetic diagnosis. And I would argue that number is actually higher, probably 50 to 60%, because we are not phasing any of these patients for the most part. So once we find a patient with a compound heterozygote, oftentimes we close the book and say, well, we've given you your genetic diagnosis. But that is not always the complete story, and as we're finding out, that can have severe implications, especially for moving these patients forward in gene therapy trials. The other issue is that there's a great diversity in genetic panels that are out there. For other people in different organ systems, there's no one panel that encompasses every gene for the most part. So when we first started this endeavor with our genetics department, we looked at the 3 common panels that are ordered through our university, and we found that they're all very different. Not only are there different in coding genes that are being covered, but also non-coding elements in those retinal disease genes. So you could send off a panel for a patient and get a non-diagnostic result, not because they don't have an IRD, but you just chose a panel that didn't cover that gene that they might be causative for. So as you can imagine, there are these disadvantages that exist in the system that we need to fix. And this is basically taking us from giving almost 50% of people a genetic diagnosis in inherited retinal diseases. So what are our limitations? Well, one, we're only testing coding exons for the most part. They don't encompass all disease genes in an organ system, potentially. Modifications that are very difficult. I didn't detail this, but bead capture, amplicon capture, all of these, in order to make a new gene product or gene target, it's going to take a lot of pre-sample selection to do so. It's not easily customizable. And we cannot face patients with short reads, and we've heard about this extensively from different talks, so I won't belabor this. So what's the answer? Well, the answer is, well, we can just genome sequence everyone, and we'll get wonderful results, but that's not realistic. Part of it is not only cost, but the analytical time that it would take. But today I'm going to talk about how we do have the ability with ONT to do genome sequencing in a targeted manner that allows us to not worry about pre-selection, anything like that, but use bioinformatics to target the genomic loci of interest so that we can really find these targets in a specific disease phenotype. I'm going to start by talking about a gene that we work on extensively in my lab called USH2A. USH2A can cause two very different diseases. One is isolated retinitis pigmentosa, where a patient develops blinding disease over the course of their lifetime. The other is Usher syndrome, where the patient is born congenitally with no hearing, and then loses vision over the course of their lifetime. Very different diseases, but the same gene. Now, the gene is quite large. It's almost a megabase with 72 exons, and you might imagine, well, if I run my whole genome, I'll be able to find it quite easily, but you won't, because our genome is quite vast. So if you really want to find it, you're going to find it, but it's going to be 0.01% of your total DNA. So if you are looking for someone with this potential genotype, whole genome sequencing will give you the answer, but you will spend a majority of your time, vast majority of your time, sequencing and analyzing items that you may not be interested in. So how can we do that without making a pre-selected array for USH2A or anything like that? Well, we can use ONT technology and bioinformatics that Matthew Luce's lab developed with read until and now adaptive sampling with ReadFish, where we can basically sequence everything that comes in. And then we have the ability, bioinformatically, to say, do we continue sequencing or do we not? And that decision will drive enrichment of your product only and allow you the appropriate depth of coverage to do haplotyping, variant calling, methylation calling, everything to identify disease variants. So our workhorse is not the Prometheon or the P2 or even the GridION, it's the MinION. Part of it is because that's what we started with. Now, I will say that we do have a very high-powered computer connected to it to run real-time base calling on the super accurate model in real time. But what we're able to do is, with a kind of an entry-level device from Nanopore, really drive disease diagnostics. And I think that what makes this technology so appealing, not just our lab, but any lab, is that there's a low input to get into it, and once you're in, you're able to do quite a lot of get a lot of information for your organ system or disease gene of interest. So basically, in a nutshell, what read until or adaptive sampling is doing is it's basically reading the DNA that's coming through after X number of bases. It's going to decide, is this belong to the bed file that I've been given? And if it does, let's keep sequencing it. If it doesn't, the Nanopore has the ability to reverse polarity and kick that thing out. And basically what this does is it takes you from a non-enriched channel that you can see there, where you get very low, shallow whole genome coverage, to enrichment, where we, on average, get 15 to 18x enrichment of our data. So instead of getting 3, 4 reads in a region, you're getting 25 to 30 reads, which is enough to do haplotyping and base calling and variant detection. Now, the biggest thing that this allows us to do is haplotyping. And there are really two configurations when a patient comes in with two different disease variants. They could be in cis, which means that they're on the same chromosome and they have a completely normal chromosome, or in trans, where both chromosomes are affected and likely disease-causing in an autosomal recessive patient. Now, the way to normally do this, if you did, let's say, clinical short-read sequencing, is this is a patient of ours where in black you could see they have two variants in green and red that we've highlighted. And if you really want to know if they're on opposite chromosomes, you got to find mom and dad, you got to get them to get tested. And then, if in this case you can see that yes, mom has one, dad has one, you're able to conclusively say that these two variants reside in trans. But if you did this and mom or dad did not have one of the variants, then one of them are de novo, and you cannot use familial segregation to phase. So even when you're able to get sample, you cannot always get your answer with short read sequencing. The beauty of using adaptive is that with the depth that you can get enough data from one flow cell to accurately haplotype. This is a family where two subjects are afflicted with USH2A-related disease, and the mother who we're able to sequence is not. And both subjects have two variants, both in trans, while the mother is a carrier of the small deletion that is pathogenic. The other thing is we need to understand not only if things are in trans, but how certain variants are aligned. Oftentimes, there are patients that have multiple disease variants, not just two, but three or four. And in what combination they are will drive disease. So in the top example, this is a patient with very early onset vision loss. So the central area in that bottom photograph is black because they've kind of lost all their photoreceptors in that area. And they have three variants in a disease gene called ABCA4. And over a 20 kilobase region, we can accurately phase this and say that of the three variants, the two severe ones are in trans, meaning both chromosomes are severely affected, which is why they have early onset disease. In the bottom example, this is a much older patient who has 20/20 vision and very mild manifestation of disease, despite having two severe variants and one variant that's hypomorphic, or less disease-causing. Turns out that two severe variants are in cis and the hypomorphic is in trans, which would explain their disease phenotype. And again, these are done without having to get familial data. Actually, for the first patient, they're adopted, and we would have never been able to phase them without the use of long-read sequencing. Now, there are errors that occur in short reads and long reads. And one of the things that when we first came into the space a few years ago is try to identify what those errors are and see, is long read going to give us the answers that we can get with short reads? Now, the errors are systematic in nature. In short reads, there are regions that are just not covered. In long reads, we have regions that are homopolymers that are not called asked correctly. But I think the difference to take away is that in long reads, you will still call them as homopolymers, but you may have to then figure out what the exact length is. Again, with low complexity, you don't cover reads, but Nanopore is able to solve areas that Illumina fails. And actually, when we looked at our data, because many data points to this point have said that long reads are not concordant with short reads, that's actually not true. The difference is how you align the data. So this is an example where you look at the reference and the output from Nanopore in Illumina. The output is exactly the same. The difference is how these things are called. Illumina calls it as a deletion and an insertion, and Nanopore calls it as two deletions and a single nucleotide variant. Now, at the face of it, you would say these are two very discordant data sets, but they're actually perfectly concordant. And when we look through our data and use this variant context matching method, we find that most data is concordant between Illumina and Nanopore. The cost functions are very different because we're using different aligners, but when you basically stabilize for that, you'll find that your data is going to actually be very equivalent between these two methodologies. And then actually, it allows better disease variant discovery. We have higher F1 scores with long read data, and this is all published, so I won't belabor this point. And using haplotyping, we can reassign variants from unknown significance to pathogenic and provide complete diagnosis for patients. Now, I talked a lot about a single gene USH2A, but I also began this talk by telling you that no single gene is really as clear about a path as a phenotype in retinal diseases. So we've expanded our workflow from targeting single genes to targeting every gene that we can think of to ever cause an inherited retinal disease on a single panel. This is a 373 gene panel with about 20x coverage of every gene. Those genes on the side there at the very end of the X are about half coverage, and that's how we know that we've sequenced a male. Now, not everything went perfectly when you ran it, so I'll kind of walk you through some of the things to work through if you ever do targeted paneling, is that when we first ran this entire panel, we found that the observed versus expected reads were not equivalent. And we kind of looked at these genes. There were about a very small subset of them. And in this case, for the gene NRL, which is a known disease-causing gene, there are a lot of reads that are aligning, but they're all supplementary reads. So there are different GRC38 bills that have been published, and what I encourage you to do if you ever have issues with aligning your data is to see if there's any deletions or duplications or regions that need to be masked. When we mask these regions very accurately, we can get our data exactly as we would have expected. And then we're able to actually get observed versus expected ratios exactly aligned. Now, the future, I think, is the ability to combine all this into one thing. And the beauty of Nanopore and adaptive sampling is that we're able to do this very quickly and provide a full diagnosis. So a clinical patient, if they are diagnosed with a disease, may first get panel testing, discuss the initial results, and then if they have two variants in an autosomal recessive disease, you'll then have to trace it through their family and then ultimately get to the phased result. You can imagine this can take months, especially with going back and forth with visits, getting different patient samples, redrawing samples, things like that. This is an example of a patient who came in who had just had his diagnosis that we immediately sequenced on our platform to identify two deleterious variants in trans and a known inherited retinal disease-causing gene. So you kind of eliminate these results, and you go from initial testing all the way to a phased result right away. And this can be done in a matter of days and actually in a matter of hours. So we looked back at our data post-hoc to see how quickly did we phase these two deleterious variants that are marked in red. And at the 11, 12-hour point, we had enough coverage and depth and phase to know that these two variants had been identified, and this was likely the diagnosis for this individual. Also, the beauty of this is that it's whole genome coverage of your loci. So this is a patient that was monoallelic in that they had one variant that had been called to be deleterious, but not a second. And on a single run, we could see that they harbor a second pathogenic variant in a non-coding region that would fully explain their diagnosis. And then, I'm going to end the talk by talking about expanding this beyond just inherited retinal diseases to ocular cancer. So retinoblastoma is the most common pediatric neoplasm. And in this case, it actually does matter how quickly you get a diagnosis because it dictates their care. Inherited retinal diseases are slow and progressive, but retinoblastoma poses a risk for metastatic spread. And similarly, we've shown that not only can we fish out all of this stuff with targeted testing, but we can also look at methylation signals, which can be used for a variety of stuff that we've learned about today. We can identify variants and trace it through families very easily. And because of the depth from adaptive, you can identify low-level mosaic variant identification. This patient had gone clinical testing and had the mosaic identified 8 months after initial diagnosis, which had driven his care in a completely different way that we were able to produce very quickly. So in conclusion, targeted long-range sequencing using adaptive can provide comprehensive information of your area that you want to assay. There is a rapid turnaround time compared to what clinical systems are now designed to do. And especially as clinical trials progress to therapy, whether it's in retinal disease or other organ systems, you're going to need to know what variants your patients have and in what orientation allelically they reside so that you can actually get them to the correct therapy. Now, this kind of will end with how this can also help us explain patients' phenotypes when they don't make sense. So this is what a patient looks like when they have ABCA4-related Stargardt disease. It's a very clear phenotype where if you look at the retina, you see these yellow flecks, and then the bottom black and white photo is the autofluorescence your retina produces, and it's really bright because these flecks are abnormal accumulation products. These are two patients that do not look like that at all. You don't have to be a clinician to kind of see that, but they both came back with variants in ABCA4, known to cause Stargardt's disease. Now, you can convince yourself that maybe this is an abnormal way that the variant is causing disease, and this is a atypical presentation, but the other answer is it's not the answer. And very conclusively with long reads, we can show in this patient, for example, that both variants reside in cis. And the second haplotype that we can at least tell does not harbor any pathogenic structural variant, methylation signal, anything. And it really is up to you then to investigate what else could it be. And we have some hits that might explain their diagnosis in the eye and extraocular that might be more related based on long read sequencing. So I think in the future, the ability to enrich for a single complex genomic region or multiple genomic region will really help us identify these class of diseases that are undiagnosed and expand that profile of 50% diagnostic rate to much higher. We may never get to 100%, but being able to get that over 60, 70, 80% would really help out understanding of what patients have and ultimate disease diagnosis. As we talked about, we can expand this from single to multiple genomic loci. All it requires is a bed file. So it really just matters what you want to sequence, and that can be easily customized for your needs. And I think the biggest takeaway is that the sufficient accuracy that you can get from a MinION makes this accessible to any lab. When we first started out, we had one MinION, and now we've expanded to many more, and we have a grid ion. But even with one MinION, you can make a real difference in understanding variants, allelic heterogeneity in chromosomal architecture. With that, I just want to end and thank the people in my lab, especially Kenji Nakamichi, who's here today, who's really driven the bioinformatics and the adaptive sampling in the lab and really made it accessible to us and hopefully a lot of other people and our funding. So with that, thank you very much.